Welcome to The Explainer. Today we're doing something pretty incredible. We're essentially flipping open a mechanical pop-up book of monsters to discover how humanity can finally cure 10 prehistoric pathogens. I'm talking about ancient diseases that have literally stalked us for millennia, surviving absolutely everything modern medicine has thrown at them. But as you'll see, we're about to turn the tables using some seriously cutting-edge biophysics.
So, here's our roadmap for today. First, we'll look at the prehistoric 10. Then, we'll systematically dive into the genetic hijackers, the barrier builders, and the untouchable monsters, before wrapping it all up with the Extinction Treaty. Let's get right into it.
Section 1. The Prehistoric 10 and the Weapons of Biophysics.
Now, these 10 pathogens have survived for so long mostly because blunt, traditional biochemistry completely fails against them. When a bug hides inside your cells or builds a wall around itself, chemical drugs just bounce right off. But we've got 3 phenomenal biophysical tools now. First up is genetics. Think of this as our molecular scissors, or really a biological search and replace. We're using advanced CRISPR, prime editors, and meganucleases to safely rewrite the pathogen's DNA while it's hiding inside us. The real genius here? We can do this while completely avoiding those dangerous double-strand breaks. In the past, snapping both strands of DNA could cause chaotic, unintended mutations in our cells, but this new method is perfectly surgical.
Next, we have photonics. You can think of this as a literal molecular scalpel. We're talking about femtosecond and plasmonic lasers that use coherent resonant frequencies to mechanically shatter specific bonds in the pathogen. If that sounds like heavy sci-fi jargon, just picture an opera singer hitting exactly the right pitch to shatter a wine glass across the room. The laser vibrates the pathogen's unique structure until it literally falls apart, all without generating any heat. So, your healthy tissue stays completely untouched.
And finally, acoustics are molecular opener. By using focused ultrasound and microscopic bubbles, we can gently buzz open formidable physical walls in the body, like the blood-brain barrier or the dense shell of a cyst. We just open these fortresses transiently, just long enough to let our genetic and photonic cures slip right through the cracks. It's an absolute game changer.
Moving on to Chapter 2, the genetic hijackers, pathogens hiding in our DNA and cells. We're starting with the viruses and parasites that ingeniously hide right inside our own cellular machinery, which makes them completely invisible to our immune system. So that brings us to a core problem. How do we delete a monstrous invader that has actually woven itself into our own biological blueprints? I mean, if you just blindly chop it out, you risk mutating the host, right? We have to be impossibly precise.
Our first monster is HTLV1, an ancient retrovirus. The devastating issue here is that it maliciously weaves its own cancer-causing code directly into our T cells, literally turning our immune defenders against us. To fix it, we execute a brilliantly coordinated 3-step heist. Step 1, we use base editors to safely and permanently silence its cancer-causing tax genes without breaking the host DNA. Step 2, we use focused ultrasound to deliver this cure straight into the lymph nodes where the virus is hiding out. And Step 3, a dual isotope PET scan acts as a spotlight, identifying any remaining viral sanctuaries so we can target them directly.
Disease number 2 is Epstein-Barr, the virus that's in almost everyone. Its main defense is extreme stubbornness. It hides in our B cells in a deeply dormant, circular DNA state that standard drugs just cannot detect. The fix here is a really clever shock-and-kill tactic. First, we unleash a customized CRISPR attack tailored specifically to chop up the virus's circular episome. Next, we use a tool called dCas9 to aggressively force the virus out of hibernation. Once it's exposed and reactivating, a standard antiviral like ganciclovir can easily sweep in and efficiently wipe out those exposed cells.
For disease 3, the creeping herpes virus, the main issue is location, location, location. It hides deep inside our nerve ganglia, protected by a tough physical nerve barrier that drugs just can't cross. So our fix starts with a quick chemical pulse, an HDAC inhibitor, to temporarily wake up the sleeping virus. Then we use MRI-guided ultrasound to gently massage open that protective perenurial nerve barrier. Finally, we slip in AAV9 meganucleases. You can think of these as highly specific DNA shredders that permanently destroy the accessible viral code right there inside the neuron.
Disease 4 is varicella zoster. The terrifying thing here is that it gives you chickenpox as a kid, goes dormant for decades, and then strikes twice, coming back as excruciating shingles later in life. To break this agonizing loop, we deploy a targeted CRISPR tactic to permanently knock out the exact genes, ORF61 and 62, that allow it to reactivate. But we don't stop there.
we give an mRNA boost to call in local immune reinforcements, strengthening the tissue-resident memory T cells. Then, we use ultrasound to deliver a massive dose of acyclovir straight into the ganglia. Boom, loop broken.
Rounding out our hijackers is disease 5, malaria. The issue with this parasite is its sheer scale and history. It's so ancient and devastating that it actually forced human evolution to rewrite our own genes to try and survive it. Our modern fix is incredibly multi-layered. First, we preemptively base edit a patient's red blood cells to make them naturally resistant. Then, we fire a highly specific diagnostic laser that targets the parasite's own iron crystals, called hemazoin. The laser instantly pops the parasites from the inside out, using their own biology against them. Finally, we use acoustic liposomes to trigger the release of primaquine deep within the liver to clear out any last dormant stragglers.
Let's flip the page to Section 3, the barrier builders, pathogens hiding behind physical walls. So, here's the billion-dollar question. How on earth do you deliver a cure when the pathogen builds a dense, impenetrable fortress right inside your body? It's a massive challenge. If the drug can't physically touch the bug, the disease persists forever.
Let's look at disease 6, latent TB and toxoplasma. The fundamental problem here is that they build literal fibrous fortresses, like granulomas or cysts, in your lungs and brain. Traditional drugs just bounce right off these walls. So the fix is a coordinated biophysical breach. First, we use acoustic shock waves, a process called hysteresis, to safely fracture those dense walls. Then, we use an ultrasound technique to create an acoustic fountain that actually mechanically pumps drug-loaded liposomes deep into the fractures. Once they're inside, we fire a photonic laser that specifically bursts those liposomes, flooding the fortress with the cure from the inside. It's like a Trojan horse, but on a microscopic scale.
Disease 7 is rabies. The issue here is beyond terrifying. It's historically been a 4,000-year-old death sentence because once it enters the central nervous system, it locks the blood-brain barrier behind it, keeping all treatments out. To fix this, we use focused ultrasound to temporarily and gently massage open that blood-brain barrier. Through that microscopic opening, we flush a specialized cocktail of Symbal-1 antibodies and AAV9 siRNA into the brain. Finally, we hit the brain with a highly calibrated combo of femtosecond lasers and acoustic disruption. Because the viral envelope has a different physical resonance than human brain cells, this combo precisely bursts the virus into pieces while leaving the surrounding brain tissue completely unharmed. Disease 8 is the guinea worm, historically known as the little dragon. The horrific issue here is its life cycle, which usually ends with a 3-foot worm painfully emerging through the skin over a period of torturous weeks. No thank you. Our fix here is to detect it early and decoy it. We use a simple urinary exosome test to detect the worm's microscopic traces months before symptoms start. Then, rather than painfully pulling it out, we coax it out gently in hours using a combo of acoustic cavitation and chemical pheromones. And to make sure nobody else suffers, we deploy a self-limiting daisy chain gene drive into the water supply. Think of a gene drive like a genetic timer. It spreads a sterility trait that safely crashes the local parasite population, but has a built-in expiration date so it doesn't spread globally. Let's move to Section 4, the untouchable monsters, pathogens with no DNA or massive genetic decay. These diseases completely defy traditional biological rules. They either have no DNA at all, or their DNA is so wildly decayed they barely function like normal bacteria. This perfectly illustrates the ultimate medical paradox. How do you kill a disease that isn't even alive and has absolutely no genetic code to target?
The issue with disease 9, Kuru and prions, also known as the laughing death, is the sheer horror of how it operates. A prion isn't a virus. It's literally just a single rogue protein that convinces your healthy proteins to fold incorrectly, creating a deadly domino effect that destroys the brain. Because it has no DNA, antivirals are absolutely useless. So our fix relies entirely on starving it and breaking it. First, we use epigenetic silencing to permanently turn off the human body's supply of normal PRP proteins, literally starving the disease of the dominoes it needs to copy itself. Then, we flood the brain with ICSM-18 antibodies to trap any existing misfolded templates. Finally, we apply a resonant ultrasound frequency that mechanically unzips and shatters the toxic amyloid fibrils so the brain can just flush them away.
Finally, Disease 10, leprosy, the flesh eater with no pain. The issue here is extreme stealth.
Its heavily decayed genetic code allows it to hide in plain sight by completely shutting down the local immune system, slowly eating flesh while turning off your pain receptors. The fix is highly active. First, we use a light-activated drug, verteporfin photodynamic therapy, to aggressively unmask the stealthy bacteria, triggering an all-out immune attack. We then use DMF to force the bacteria out of its metabolic hibernation so antibiotics can deal the killing blow. But the real biophysical breakthrough is step 3. We use high-resolution ultrasound to guide a regenerative stem cell transplant directly into the patient's damaged tissue, actually healing their dead nerves and restoring the sensation they lost.
And that brings us to Section 5, the Extinction Treaty, engineering the end of pathogens.
We've closed the tabs on our 10 monsters, so let's look at what this all means for the future. We are witnessing a massive medical paradigm shift right now, moving from blunt biochemistry to precision biophysics. In the old paradigm, we relied heavily on chemicals, but chemicals fundamentally fail against dormant or physically fortified threats because a pill can't always tell the difference between a sleeping virus and a healthy cell. In the new paradigm, we use resonant physical properties. We target what the pathogen physically is, its specific genetics, its unique photonic vibrational frequency, its acoustic density. By targeting the physics of the disease, we achieve perfect, uncompromising selectivity without harming the host. It's absolutely brilliant.
As the prehistoric pathogen treaty puts it, the roadmap becomes a self-sustaining, accelerating immune system for the human species. By treating these biophysical blueprints as an open-source public utility, we don't just treat individual symptoms anymore. We finally have the biophysical weapons to engineer the permanent extinction of humanity's oldest predators. So the only question left for us is, are we bold enough to ratify the treaty, close this bestiary for good, and live in a world where these monsters are just myths? That's a lot to think about. Thanks so much for joining us on The Explainer today, and we'll see you next time.